Hi, we did our paper on Dr. Laurie Ferentz's study. It's titled Pharmacore Identification and Structure Activity Relationship Analysis of a Series of Azendols as Inhibitors of Trypanosoma brucei. Some background. Human African trypanosomiasis, or HAT, is WHO designated as a neglected tropical disease, and it's caused by the parasite T. brucei. This condition is typically fatal if untreated within those first two stages. The first stage is where the parasite is in the blood and lymphatic system. And then the second stage, typically fatal, is when the parasite passes the blood-brain barrier into the central nervous system. Uh, Current treatments, there are two. One was approved in 2018. It's an oral tablet. And the other one is currently in trials, but they're complex treatment regimens. And there are also some issues with resistance, considering that this is a parasite that can mutate, as well as toxicity to humans and mammalian cells. So for this study, it's nonprofit sector. Typically, that also means limited resources. The private industry typically would not go for this area because it's a neglected tropical disease. There's not much benefit when for like cost benefit necessarily. And so for this, they repurposed human kinase inhibitors for parasitic targets and did an initial screening of GSK kinase focused collection. It's about a little over 42,000 compounds, and they identified four amino azendols as promising hits. The four amino azendol right here, it has this hydrogen that is a hydrogen bond donor. It was found to be pretty helpful in finding a hit. So the approach and methodology for this specific study, it's cheminformatics driven. They did an initial screening here. All the gray compounds are everything that they screened. And then the green ones are the ones that were actually selected to be synthesized. And they were done through Buchwald-Hartwig coupling reactions. And additionally, there were growth inhibition assays that basically just compared T. brucei to mammalian cell lines and seed and saw whether they were cidal, fast acting, all of those things. And then additionally, there was ADME profiling to figure out some more aspects of those compounds that were being studied. Their lipophilicity, molecular weight. There's more you'll see right here. These are all the ADME things that they were studying or that they tested for. This was the initial compound. This is hit one. It shows some good potency against T. brucei. You can see the PEC 50 is above 7, like it was supposed to be that targeted value. And it was shown to be selective for T. brucei over mammalian cells. Additionally, it showed moderate lipophilicity and it was fast acting, but not cidal in vitro. So there's something that needs to be changed about the compound for it to be a good hit. Additionally, they also identified off-target activity against human ROC1 kinase. So that also needs to be addressed as they go further into studying different compounds. So first, they decided to make some alterations to the pyrazole region. You can see here there's R1 and R2 positions, and those were changed with, as you can see down this column here, different things. It was found that the hydrogen bond, that hydrogen right here, that hydrogen bond donor on that pyrazole was critical, and that smaller alkyl chains were favorable. So methyl, again, being just one carbon, ethyl, propyl, two and three, and then longer chains were less favorable. Specifically for the R1 position, ethyl and propyl were favored, and again, bulkier chains were not tolerated. And for the R2 position, the methyl was favored, and again, longer chains decreased that potency.
So to pick up where Spencer left off, in this study, the 7-azandyl moiety was used as the core scaffold for SAR optimization. Replacing this moiety across a series of analogs helped probe how different heterocycles affected antiparasitic potency against T. brucei. The results clearly showed that certain replacements can retain or even improve activity, making heterocycle optimization a key driver in developing a more effective compounds. Compound 4E, where 7-azandyl was replaced with an indole, showed a modest increase in potency with a PEC30 of 6.4 compared to 4A's 4.8. This suggests that the indole scaffold contributes positively to activity. The benzofuran derivative 4S demonstrated an even greater increase, really, in potency with a PEC50 of 8.0, the highest of any in the series. This suggests the oxygen atom may serve as a crucial H-bond acceptor, and the overall dipole movement or electronic environment of the ring improves binding affinity. The SAR trends indicate that the N-alkylation at the heterocycle is generally well tolerated, as the H-bond donor from the NH is not essential. For instance, compounds that lack traditional H donors, lack a traditional H donor, such as 4S, still retain or even enhance potency. This highlights flexibility in the design and opportunities to improve metabolic stability or pharmacokinetic properties via N substitution. This data suggests that the H-bond acceptors play a more critical role in binding than the donors do. So when it comes to pharmacophore identification, to guide compound design, a pharmacophore model was developed based on known active scaffolds, specifically incorporating the donor-acceptor-donor hydrogen bonding motif from the pyrazole. So in this model, we see here, the blue spheres represent the hydrogen bond donors, the red spheres represent the hydrogen bond acceptors, whereas the green represent the hydrophobic region. The aromatic feature, the orange ring, shows the pi-bond interactions, which are common in ligand-protein binding. These interactions help orient the compound and can increase binding affinity. When it comes to benzofuran derivative 4S, its replacement of azanedol can significantly improve potency. Sorry, I have a bit of a cold here. Key properties: the PEC50 improved from a 7.4 in compound 1 to an 8.7 in compound 4S. It maintained selectivity over all mammalian cells. It eliminated off-target ROCK1 kinase activity. It also demonstrated sodality in vitro. And in vitro metabolism studies showed moderate clearance as well. So when it comes to pharmacokinetics and efficacy, a PK study of compound 4S demonstrated high exposure levels in both systematic circulation and the brain. Figure 6 from the paper shows stimulated PK profiles following IP administration in mice. And notably, 4S reached concentrations exceeding the PEC50 in both compartments, indicating its ability to distribute beyond the blood-brain barrier, which is a critical factor in treating CNS-resident parasites. The predicted therapeutic exposure target was a drug-free concentration exceeding the PEC50 for at least 8 hours. The observed plasma concentration time profile met this requirement, supporting the selection of this dosing strategy. Sorry. Based on this PK profile, a twice-daily dose of 10 milligrams per kilogram was predicted to maintain sufficient drug levels for therapeutic efficacy over a full day span, 24 hours. Mice infected with T. brucei were treated with compound 4S twice daily via injection, which is a common route for preclinical models. And again, while not a complete cure, 4S significantly extended survival in infected mice by 11 days, which indicates a strong antiparasitic pressure. It did fail to completely clear the infection. Not comply. It failed to completely clear the infection, which is conclusive that basically the efficacy profile indicates trypanostatic behavior, meaning that the compound inhibited parasite growth rather than killing them outright. And this distinction is important for evaluating mechanism of action as well as future optimization strategies. So finally, we'll end with mechanism of action studies. Isolate the molecular target of these compounds. The chemoproteomics approach was used to profile both human and T-Brucey kinases. This technique allowed researchers to map compound protein interactions across a panel of potential enzymatic targets in a cell-relevant context. One key hit from this profiling was arginine kinase, a parasite-specific enzyme involved in ATP buffering and energy metabolism. This finding prompted further biochemical investigation into whether the enzyme played a functional role in compound activity. Sorry. Only compound 4C and 4M showed any significant in vitro inhibition. Despite the identification of arginine kinase as a binding target, only two compounds, 4C as well as 4M, showed measurable inhibition in enzyme assays, including limited interaction across the full series. There is a disconnect between the biochemical inhibition clearly and the potent antiparasitic activity observed in cells. Even for the most potent enzyme inhibitor, compound 4C, the IC50 was around 1.3, whereas its EC50 in T-Brucey cell assay was the low nanomolar range, which is a difference of over 1,000-fold. This large disparity further suggests that enzyme inhibition does not drive cellular efficacy at all. Altogether, this data suggests that the arginine kinase may be a non-essential or secondary target, that the compounds likely engage with multiple targets or pathways in a parasite.